Hey y'all, it's Miss Lynn here. I am so excited to be back with y'all. And today we are going to start our first review enrichment lesson on viruses and bacteria, something that we are obviously dealing with at this current moment. So I'm excited. I'm going to go over a couple announcements in the next couple pages. Obviously, we are not in the classroom right now, but I'm so glad that each and every one of y'all have logged on to listen to this mini lecture today. This is considered enrichment still, so just so y'all know. Today, if you look at our nifty little calendar, we are doing viruses review, but tomorrow I will not be posting a lesson. Tomorrow I will have a Zoom meeting, if you would like to join, at 9 a.m. to 10 a.m., and that is for any questions, concerns, if you just want to call and chat. I would love to see all of your faces. And then the following day, we will start with a plants review, and we'll do that rotation of having a lesson posted with a worksheet, an exit ticket, followed by a Zoom meeting, practice review, study the following day, just like you would have a normal AB schedule. These videos will not be 90 minutes. It will not be a typical class period. I will try to keep it 30 to 20 minutes. And if I go over, I will always apologize. But just to let y'all know, I will try to keep it in that time frame just to respect your time and your access to whatever internet technology you're using as well. I posted this calendar on your Google Classroom, as some of y'all might have seen and known. Somebody did ask, I made this schedule all the way up until April. Does that mean we're going to be out of school until April? I have no idea. I am just preventatively laying out some lesson plans for the future. I will update the information as soon as I know, but as we're going to talk about today, it might be a little bit longer than we think. And so we're going to talk about that as we talk about viruses today. But like we were saying, today we're not only talking about viruses, but we're also talking about bacteria. So we still have a statement of inquiry right here. And our statement of inquiry is, I would normally have a student read it, but I'll go ahead and read it. It is, I will be able to describe the characteristics of bacteria and its importance. And we're also going to be able to understand the structure of viruses, describe how they reproduce, and also describe the role of viruses in causing diseases like the ones that we're dealing with right now. So to start off with bacteria, we do know, if y'all did not remember, bacteria are considered prokaryotic. And if y'all don't remember that little trick, pro equals no. Does anyone remember what no stood for? It means no nucleus. And so all bacteria are prokaryotic because they do not have a nucleus inside them. They come in three different forms: rods, which as you can probably see from this picture, they are a rod shaped. Spiralia, which are circular shapes like this, as well as spherical, so almost like a spiral or a sphere. So just so y'all know, those are the three types of bacteria shapes. Some bacteria cause diseases. If you've ever had a bacterial infection, you know this. They actually invade the tissues of your cell and can also make toxins. A toxin is a poison released by the organism. Now, there are some diseases caused by bacteria. Some that y'all may have experienced as well is things like acne. I know most of us have experienced acne before. That's caused by an increase in oil production in your skin, and paired with that, this bacteria will actually cause acne. There's other ones that you might not have heard of, like anthrax. That's actually where you can inhale endospores. And you're inhaling the endospores of this bacteria, Bacillus anthracis, and you'll have these symptoms of fever, trouble breathing. Have you ever heard of Lyme disease? That can also be caused by bacteria. Tetanus, if you ever stepped on a rusty nail, a wound contaminated with soil, anything rusty is typically can cause tetanus, and it will have this bacteria in it, and it will cause severe muscle spasms, fever, and even something called lockjaw, where your jaw locks up. And then the last one that I want to talk about is tooth decay. All of us should be brushing our teeth because Streptococcus mutans are going to be large bacteria found in your mouth. So when you brush your teeth every night, you're getting rid of that. Now, how do we treat bacteria? Obviously, with that blast bacteria that we talked about that causes tooth decay, we can brush away every single night. But let's say it's a bacteria that we can't just brush away like on our teeth. We treat bacteria diseases with antibiotics. Antibiotics can actually stop the bacterial cell wall from forming, which means the bacteria can't replicate. And if it can't replicate, then we've stopped it in its tracks. It can't go on to reproduce, and our immune system can eventually attack it. Now, if you did know anything from our last discussion on viruses, antibiotics do not work on viruses. Do not. So how do we fight off viruses? That's what we're going to talk about. Prevention is the best method to fight bacterial diseases, which means just preventing getting any of these bacteria. A good way to do that is, like we've talked about in the recent news, washing hands. Now, over time, bacteria can actually evolve resistance to these antibiotics. So let's say that you have a bacterial infection and your doctor tells you to take 10 days of this antibiotic, okay? But let's say after day 4, you're like, oh, I feel good. I'm just not going to take the rest of my medication. Well, your bacteria that caused whatever disease that you had can actually become resistant to this. And we're going to talk about this. So if you don't use your antibiotics correctly or you overuse them, these bacteria can become resistant. So antibiotics must be used properly or they will stop working altogether. So in that example that I was talking about, let's say your doctor prescribes you 10 days of an antibiotic. And in the beginning, yours looks like this. You've got lots of germs, and some of them are drug resistant. So we've got normal bacteria here. So all of these are our normal bacteria. We've got one that is drug resistant, okay? As soon as you take that antibiotic, it kills all of the bacteria that were normal bacteria. But what does it do to this one that's resistant? Well, it lived, okay? And it's going to go on to continue to grow and take over. Now, did you get rid of all those bacteria like you were supposed to? No. So this actually happens when people, let's say, take only 4 out of the 10 days of antibiotics, and they're like, oh, I'm feeling better, I don't need to continue taking it. What ends up doing is it kills a bunch of these normal bacteria, and one bacteria can end up becoming resistant. Well, that resistant bacteria is going to continue on multiplying, and that disease has not gone away. So that antibiotic has actually become useless. So really quickly, we're going to have a quick bacteria summary. We learned that they are prokaryotic, and if they're prokaryotic, prono means they do not have a nucleus. No means no nucleus. We also learned that they are treated with antibiotics. And they reproduce very fast. We learned some examples of what diseases are caused by bacteria, such as acne, tooth decay, different respiratory diseases. And so now we are going to move on to viruses, which is what y'all are here for. So I'm going to start off with introducing to y'all multiple types of viruses. So this first one we have is called adenoviruses. It causes actually respiratory infections. So what we are dealing with in the world right now is the coronavirus that actually can end up causing different diseases called MERS. I'm going to write that on the board right here. MERS and SARS. We're going to talk about those a little bit later, MERS and SARS. Adenoviruses can also cause similar symptoms. We discussed this the last time I taught viruses, but also any type of flu viruses. If you did not know, this is actually what the flu virus looks like. Pretty crazy and cool. And this is very similar to the coronavirus structure that we're going to talk about in a little bit. Viruses can also form lesions on your skin. If you have an example of a virus that does that is smallpox. If you've ever seen smallpox pictures, it leaves small lesions or small bumps on the skin. This is also caused by a virus. And the last one that I wanted to talk about is also viruses don't just affect humans. This is a virus that actually attacks plants. So TMV is tobacco mosaic virus. It's the first virus ever discovered. And if you notice it, this is a tobacco plant. A is obviously uninfected. B is after a couple days and C is after about a week. It caused all these little small lesions on the leaf, and they're little holes inside the leaf that are going to end up killing this plant. So obviously viruses can not only affect humans, it can affect all living things, including other animals. So just like y'all might have heard, the coronavirus started in bats. And so we're going to talk about how all these different viruses can actually come from different animals as well. So when we talk about viruses, what do you know about viruses? We've actually already had a lesson on viruses. We've already been tested and quizzed on viruses. So everyone at home should be shouting out the answer to this question. Are viruses alive? I'm going to give y'all a couple seconds to think about it. The answer is no. Everyone should have shouted out no with their computer, okay? Viruses are not alive. Now I'm going to give you 10 seconds and I want you to think, why are viruses not alive? While you're thinking about this, think about what makes things alive. Viruses, if you did not remember, are disease-causing agents. Any disease-causing agent can be called a pathogen. So that could be a virus or like the bacteria that we talked about that infect your body. We call those pathogens. So viruses are also just pathogens. But if y'all were able to come up with some reasons that viruses are not considered to be alive, hopefully you came up with some things like they can't grow. That's a typical one. Humans grow and they get larger over time. Viruses cannot grow. They cannot reproduce on their own. Like we can actually reproduce on our own. They cannot. They need a what? Yep, you're right. They need a host. And they do not maintain homeostasis. So they can't maintain the internal balance inside their bodies. And they are not made of cells. They are made of only proteins, okay? Now, if we go into the structure of viruses actually have a pretty cool structure. And they're very, very simple. They only have 3 things. I'm going to give y'all a second to think about the first thing. Y'all can actually already look at our picture over here and see it. What is inside this circle over here? That is our genetic material. Now, our genetic material can be one of 2 things. And hopefully y'all are already shouting it out. But our genetic material can either be DNA or it can be RNA. Can there be DNA and RNA in our virus? No, it has to be one or the other. Good job. The second part of our virus that's important is this thing right here. It's going to surround and protect our DNA, and it actually starts with a C. If you did not remember what that is actually called a capsid, and it is the protein coat that surrounds and protects the DNA or the RNA, our genetic material. Because that's the most important part of our virus. We want to protect it. Okay, and the last thing that we've got to talk about is this thing that allows our virus to attach to the cell. And that is that big G word. That is called a glycoprotein. And that is actually a protein that attaches to cell surface and allows the virus to enter into the cell. Viruses are classified by their shape of their capsid and whether they use DNA or RNA. So what we mean by the shape of their capsid, for example, HIV, their capsid has a circular shape, as well as hepatitis B, influenza, herpes, and papillovirus, and rotavirus. Now, this bacteriophage, for example, has a hexagonal capsid shape. The Ebola virus almost has a spiral capsid shape. Adenovirus has a different geometric shape. And then this rabies virus has almost a cylindrical shape. So all of these are going to determine the name and the classification of the virus. And also, they all differ in whether they have DNA or RNA. Okay, we also talked about the different life cycles that a virus can have. The first one we're going to talk about is the lytic life cycle. Remember, lytic is a short, quick name. It's a short name, so that is actually going to be the quick or the faster life cycle. The first step of the lytic virus is the virus is going to inject its DNA into our host cell. So if I was to draw this first step, this is my cell. Let's say this is a virus, and this is my virus injecting its DNA into the host cell, okay? Once it injects its DNA into the host cell, or the virus itself could entirely go into the host cell, the cell will actually think that DNA is its own DNA, and it will actually read the DNA through transcription, and it will translate the DNA into different types of proteins through translation, and it will actually begin to replicate the viral DNA, and it will also replicate all of the proteins that make up the virus. So as this DNA is being read by the cell's machinery, it's going to start making different parts of this virus, and it's going to make up the DNA. It's going to make more copies of that DNA, and it's going to make all these different components of that virus. And eventually, it will reassemble those different components into new viruses inside of the cell. And eventually, it will make almost 1 million of these new viruses. And when there are too many of these new viruses inside of the cell, it will actually burst open the cell, and those viruses will leave and go out and infect other cells and do the same process all over again. Now, like we said before, lytic is shorter than lysogenic, so it is going to be our fast-acting or our quick virus that happens, which means it actually will show symptoms between 2 and 14 days after it infects somebody or another living thing. Now, what else has a 2-week incubation period that we are learning about in the news right at this minute? Yep, you're right. This is also the incubation period of the coronavirus that we are hearing so famously about. So the coronavirus is actually also a lytic virus, which means it can cause the death of the host cell, but and it can be very harmful, but your body can also fight it off through the immune system. The other life cycle that we talked about was lysogenic. Remember, lysogenic is the longer name, so it takes a long time for it to actually show up and occur in your body. In this example, we have this picture over here. The viral DNA, which is all the way up here, gets injected into the cell, and it actually will basically enter itself into the cell's DNA. So that's what we call a prophage, okay? A prophage can actually exist in the DNA of the host cell. So this prophage is made up of the viral DNA. That's important to remember. Okay, a prophage has the viral DNA, and it can insert itself into the host cell's DNA. And it can live in the host cell's DNA for generations without becoming active. And that means that that cell is going to continue to replicate over and over and over and over and over again. And at some point, that DNA will actually be activated, and it will enter the lytic cycle at that point, and it will continue with the cycle that we just went over. As you could tell, the host cell was not destroyed with the lytic cycle. Viral nucleic acid, the DNA of the RNA, joined with the cell's DNA. It actually inserted itself into that DNA. And again, the onset of these symptoms are not going to happen for months or even years, which is why we say the lysogenic is going to take a long time to pop out and begin the lytic cycle. Up next, we are going to talk about specifically the coronavirus, the thing that we have been hearing so much about. Hopefully I can answer any questions that you still have, and maybe you can educate someone in your household a little bit about the coronavirus and how you can also take preventative measures in this time period. So quickly, these slides are very dense with a lot of information. I wanted to make sure that I could give you the most up-to-date information that I could find. And I have compiled this PowerPoint along with some other teachers to bring you the most up-to-date information. So if you have any questions about it, I have resources where all of this information came down at the bottom of all of these slides. Feel free to pause the video, read through this whole slide. But so this video doesn't take 40 minutes, I'm going to just read the highlights. So again, the human coronaviruses were actually first identified in the mid-1960s, okay? So these are not new. But just so y'all know, there are 4 main subgroupings of coronavirus, and these different groupings are based on the different structure of whatever they look like. So their structure is going to determine one of these 4, okay? Now, we have, what you can tell here is 7 different coronaviruses, and this 7th one that we have all the way down here is actually the one that we are dealing with right now. It is SARS-CoV-2. We call it COVID-19 in the news, if you've heard that. And that is the novel coronavirus causing why we are not in school right now. Like I talked about a little bit, the coronavirus name is based on its structure. Okay, that's important. We name viruses based on their structure. So this picture on the right is actually the structure in a picture of the coronavirus. Okay, they do not contain DNA. They contain the other one. It's single-stranded. It contains RNA. Again, RNA is a single-stranded nucleic acid. That's important to remember, okay? Single-stranded nucleic acid. So they also have an envelope that we're going to talk about, and it is characterized by club-like spikes. So if we see, it looks like almost like a club, like it is projecting outward, and that's actually a really important part of the coronavirus that we're going to talk about. Okay, so what's actually unique about the coronavirus structure is that the virus envelope is derived from the host cell's membrane, okay? So once this virus is created within the cell, so we're going to say inside cell, this virus will actually go to the edge. It'll move to the edge like over here. Again, this is RNA inside, so it's just a single strand. And over time, as it's moving outward, it will actually grab part of this membrane. This is the cell's membrane. So actually what is surrounding the entire cell, the virus will move and grab part of the membrane and take it with it. So as it's moving out, if I was to draw my little virus here, it would have actually taken, I'm just going to draw little red dots to represent that membrane. It would actually look like this once it leaves. Okay, so it actually took part of the cell's membrane with it, and it enveloped around it. Okay, so therefore the envelope is actually made of phospholipid molecules arranged in a circle. Because the cell membrane is fluid, which means it moves around, kind of like that, the membrane is able to form around the budding particles. And so this is going to be really, really important when we talk about the preventative measures that we can talk about. But again, this enveloped virus, why is that moving? Again, this enveloped virus are surrounded by a phospholipid bilayer that is from the host cell. So if y'all remember, this is actually what our cell membrane of a cell looks like. And we talked about this a long, long time ago, all the way back in September. But it ends up with something that looks like that. And it's made up of all these circles, we call them heads and tails, of the cell membrane. And that phospholipid bilayer membrane is actually surrounding the entire virus itself once it leaves the cell. Okay, so that's really, really important. So the New York Times actually put out an article describing how the coronavirus hijacks your cells, or how it enters your body and what happens after it does. And so I put those 6 steps right here for y'all so you can kind of see it. The virus will enter your body through your nose, mouth, or eyes, and then attach to cells in your airway that produce a protein called ACE2. Okay? The virus will then inject the cell by fusing with its lipid envelope with the membrane of the cell, and once inside, the coronavirus releases its RNA. Again, we talked about how the coronavirus has RNA. Okay, so it'll release the RNA right here. You can see it. Once its envelope has fuses with the cell membrane. The injected cell's ribosomes translate the RNA. So we talked about that, translating the RNA, and make the proteins that will keep the immune system at bay, which means they'll keep it from intervening, and help assemble new copies of viruses. So that's, the cell is actually helping make new copies of the virus. The injected cell makes new spikes, if you see here, they're new spikes, and other proteins that will form more copies for the coronavirus. The new copies of virus are then going to be assembled, carried out to the outer edges of the cell, so the cell's doing all of this heavy lifting and work for it. And then each infected cell can release millions of copies of the virus before the cell finally breaks down and it will eventually die. So the cell may inject nearby cells and end up in a droplet that escapes the lungs, and that's how it's actually transported to someone else. So if you sneeze and your body has just released 1 million new viruses, some of those viruses might actually end up in a droplet and be transported to other people. Now, one question that I got a lot in class was, how do viruses form? And to be honest, I didn't know the answer, so I had to do some research. But viruses actually undergo evolution just like everything else. And it actually has happened according to natural selection. So again, that theory of only the most fit survive, well, only the best viruses continue on. And they actually mutate and change just like we do. And that was something that was a foreign concept to me, but please go to this website down here to read more about it. RNA viruses have a high mutation rate, which allows for very, very fast evolution. We talked about evolution happening over millions of years. Well, guess what? Viruses can evolve a lot quicker than that. When 2 viruses infect a cell at the same time, they might actually swap genetic material, mix their virus DNA, and actually become a new virus, kind of like we have in this antigenetic shift, which is quite strange. But that's how we also get new strains that we've never seen before. So like I was saying, viruses can mutate and evolve and change over time. And this coronavirus that we are seeing in 2019 is one of those that has mutated and changed over time. Right here, I've even included a little graph from Science magazine, and the URL is right here down at the bottom. But this graph basically shows us the mutation rate, so how often it mutates. Up here, we have a higher mutation rate, and down here we have a lower mutation rate. And then it also has the genome size. So over here, higher eukaryotes, that's who we are. We are a eukaryotic cell. And we have a very large genome. It's talking about our DNA or our RNA. We have a long, long genome. And so we actually don't mutate as rapidly. We're down at the bottom of the chart as something like a virus, which doesn't have a very large genome. If you see that, it falls right here, about 10 to the 4, which just means 10 with 4 zeros after it. So that 100,000 base pairs versus our 10 to the 9 over here. So because it has a smaller genome, it is actually going to mutate a lot faster than like us, for example. So especially these RNA viruses like SARS-CoV-2 are going to mutate rapidly. Really quickly, has this happened before? Yes. I'm going to leave this chart up here for y'all. I'm going to erase this slide. Pause it if you want to. I'm trying to beat under 30 minutes. But no, this has happened in 2003, 2013, and now 2019 is when we've seen this coronavirus happen before. A quick note, not to shame anyone who gets the virus. They cannot control what kind of infectious disease they have. A quick plug to prevent by washing your hands, because soap will actually dissolve that phospholipid envelope that we were talking about earlier. Specific to this coronavirus, not all viruses, but specific to this coronavirus, it will actually dissolve that envelope, which is why hand washing is the number one most published way of preventative measure.